awareness of this chapter, then we have to take a different way of teaching. If you know something, then we can fast forward and talk about the important aspects. Okay. So I will begin like how it is normally. So if you feel that it's slower and if you want it quite faster because you already know the chapters, I mean like you are aware of this topic, then we can go for the important parts. All right. So today's we are starting a new chapter, that is photosynthesis in higher plants. So this is a doorway that opens us to discuss about plant physiology. And physiology is beautiful. Like you really get to the mechanics of how things work. For example, if I talk about humans, uh, and if we talk about humans, what do we see? We just see the morphological characters like the number of hands, the number of fingers in each hands or the legs, how many bones are there, you know. So some of the things that you can observe visibly, which are called as morphological characters, and what is within this morphology, you study like, okay, if you take digestive system, you talk about the presence of oral organs, and then you talk about esophagus, stomach, large intestine, small intestine, and rectum. So these naming the parts would be naming the parts and understanding the locations of the part would be just anatomy. And physiology is something you talk about the mechanics of how things work. For example, why do we chew food, and why the mastication happens in such a way, and why a mouth produces saliva, and what is the role of saliva. Okay. So understanding why they are there and what is the role of them, in our body or in our in our existence. So that's what talks about physiology. Physiology is quite deep, and understanding how really they're able to survive, or an organism is able to function. It's just like studying the engine and the engine mechanics of a bike or a car. Okay, it's wonderful. Some of the people, what I see that is some students are very much happy with animals and they're connected to animals, and some students are quite connected to plants. So it's there is always a difference, and some people feel easy with both plants and animals. So there is a difference. So I will be neutral in making you understand. I want you to just relax and absorb whatever the facts that is coming out of my mouth, and don't overthink about the details, the complex words and the complex descriptions that are given in the textbook. We can simplify always, and I will try to simplify as much as possible so that you will be able to understand. Okay. So coming to the photosynthesis in higher plants. So we are talking about, I did not define, right, what is photosynthesis. You've been studying that photosynthesis is a process of manufacturing of food by the green plants using the sunlight. So this has been a very, very basic definition that we have come across throughout all our academic journey. Okay. So now, first of all, why photosynthesis? Photo is light and synthesis is using light. We are synthesizing something. So generally, what do we synthesize in photosynthesis is carbohydrates, right? For example, if I eat anything like a bread, rice, wheat, anything I eat for my food, eventually what do I need is energy. Similarly, plants need energy to survive, and do all the biological functions like every other organism does. Like how we circulate things across our body, plants also do that. How we reproduce, they also produce the pollen as well as they produce the egg. They also fertilize, and they also manufacture fruits, manufacture scents to attract things. So there is a different biology that is happening in the whole plant community. For all that, they require energy, and the major source of energy for all living organisms have always been carbohydrates. I'm not telling it's the only source of energy. It is one of the major source, and it is an immediate source of energy for our body. Carbohydrates, right? So how do they derive carbohydrates? And it comes through sunlight, water, and carbon dioxide are some of the raw materials that are used by the green plants to produce this carbohydrate. Okay. So it's an androgonic and anabolic process. Anabolic process, you know that it is a constructive process. All right. Now, the question for you is, why do plants have to choose this way of life? For example, if you look at us, we just depend on food for other organisms, right? Some are herbivore, some are carnivore, some are omnivore. So something like this. We have different ways of gaining the energy, but we are not self-sufficient. That means we require some others organisms help to survive. That's why we are dependent on some of the organisms. So if you look like that, if you see that aspect, It's quite easier, isn't it? When food is already there and all you have to do is search your food and eat your food. So it's an easy way of life, isn't it? Why did plants choose the hard way? What is the hard way? That means they have to produce everything from the bottom, right? So they're the first source of energy. That's why we call them as producers. So why did they choose? Did they have a choice? If you look back when the earth formed, when the earth was created, right, according to science, according to the astronomy. So we go back almost, basically when earth took a shape, it was around 6 billion years ago. From then, if you see that we don't have any living, or there was no life on this rock. It was just a barren rock, and it was completely, messy planet. And then it took several billion years, like 2 billion years of only life started. If you see, life when it originated, for the life to continue and to do its process, some energy source was required. And the major source of energy that all these organisms, or all the cells we can tell, are the cells that emerged first, the first organisms we are talking about. When they emerged, the only source of energy they had was light. Okay, I will mention here. So either they had the energy source as light, or they had the chemicals. Okay, chemical energy, correct? No, light or chemical energy was the only source of energy that was available for the life to thrive or to do its activities. Okay, so that is why plants or any other any organisms that you see, who have come first, they didn't have a choice but to become self-sufficient, which we call them as autotrophs. So plants are a type of autotrophs where they are photoautotrophs because they use photon is light. So photoautotrophs, that means these are the organisms that use light for the synthesis of food. Similarly, we also have chemoautotrophs. So now, if you look at the whole process, what you see in photosynthesis is, of course, they produce, uh, you know, energy in the form of carbohydrates, and to do that, they require sunlight. We know that, and carbon dioxide is also required. And by now, we have the simplistic knowledge about how the simple photosynthetic reaction happens. So if you write an equation of how the photosynthesis is, we can write like carbon dioxide plus water in the presence of light with some of the cell organelles like chlorophyll and chloroplast. Basically, you get carbohydrates, water, and oxygen as a byproduct. So now you know that oxygen, how important it is for our survival, right? So if you ask me, there might be sufficient amount of oxygen back then, right? No, not really. So because of this photosynthetic organisms only, in our atmosphere, the amount of oxygen or the concentration of the oxygen increase by several folds over several millions of years. Okay, so let's see. Now, uh, we know that the raw materials for the photosynthesis in all organisms would be basically they require carbon dioxide. Carbon dioxide is must for any organism, whether it is a chemoautotroph, whether it is a photoautotroph, it doesn't matter. Carbon dioxide is a must. And next goes is the water or any of the electron donor. We call it as an electron donor. For example, if you take water, we tell that water is required for plants growth, right? So water directly involves in the process of photosynthesis. And what is the role of water? Water acts as electron. I hope you can see the ink color. If you cannot see the ink color properly, please let me know. Okay, they are what? Electron donors. Similarly, it's not just water that can take up this responsibility. Anything with H2 plus A, I'll write it as A. A can be your sulfur, for example, you can talk about sulfuric acid or nitric acid, okay, or H2S gas. So they can break this bond between the hydrogen and the corresponding element. For example, there is a bond between hydrogen and oxygen. If you break this bond, electrons are going to be released. So using these electrons, what we do is, I mean, like what plants do is they construct, glucose. And finally, byproducts would be water. And, in case of sulfur, it would be sulfur. If it is H2S, it would be sulfur. If it is water, it would be oxygen. So this would be the waste. And this waste, you know how we utilize it. Okay. So now, before we head on to the details of the other aspects of photosynthesis, I want you to note clearly that there is one particular page in your textbook, okay, And that page would be, let me check, it should be around, guys, the first page, the very first page. And if you see there, you have something called as early experiments in understanding photosynthesis. So we know that understanding any process was not one step. It took a lot of experimentation and it took a lot of understanding, right? And it all began, okay, in the year 1933. So in 1933, what happens is Joseph, see, what I'll do is I will try to fast forward this early experiments, and I want you guys to read very clearly and make notes of each and every scientist's contribution and what they suggested. Okay, so this is your homework. You have to do this, guys. Okay, so I'll just tell it as a story. Okay, Joseph, Priestley was the first ones. What he did was, it's a bell jar experiment. It is a very famous bell jar experiment that you might have been aware. So what he does, he takes a bell jar, a jar, and then he puts a mice and a candle. So when he puts a mice and a candle, what happens when the jar is completely closed? The mice will survive for some time. After some time, what will happen? The mice will die, and candle will also go off. Now we know why. Right now we have an understanding that, oh, the jar is completely closed. If it is closed, our poor mice will not get the oxygen or the air to breathe. And for the burning of fire, what is required? Air is required. That is, oxygen is required. So that is why both of them got extinguished. And he did not stop his experiment there. He was very curious. Like, why did they grow? I mean, like, why did they die? So something like when there was a closed space, immediately both of them, like even the candle died as well as the mice died. So now what he did was, chalo, if I take another living organism, let me put a plant. So he put a mint plant in the same bell jar, and he observed what would happen. He did not expect that all of them would survive. So what happened? Something was compensated, means plants provided something that helped both the mice and the candle to survive. You understand my point? So now in this experiment, this particular person, what he did was, he was not able to completely tell that, okay, oxygen is something that is evolved by the plant. That is why the mice and candle were able to survive. He was not able to conclude. Okay. But somehow what he understood was, there was a requirement of sunlight. He thought that sunlight is required, okay, for the photosynthesis to happen. Anyways, now next what happens is, uh, the next person comes is Julius von Chauss. So Julius von Chauss, what he does is, he tells that the whole process of photosynthesis, whatever the plants are doing, they are doing it to produce glucose. So he says that glucose is something that is produced as a product of photosynthesis. And he said that starch, they produce glucose, but they cannot store glucose. Even we cannot store glucose. So they store glucose in the form of starch. Okay. And then next comes is Eagleman. Eagleman, what he does is, he takes the sunlight. I mean, he takes the light. He splits the light into whipped jar using the prism. So you know that light can be split into a spectrum. So when he split into a spectrum and he started cultivating the blue-green algae, especially Cladophora, he observed that these particular guys, they actually grew in only one particular band of light. So they preferred red light and blue light. Under red light and blue light, you could see that they were producing a lot of bubbles of oxygen. Okay, they were producing a lot of bubbles. And when he analyzed the bubbles, he got to know that this is nothing but oxygen. Are you understanding me, guys? So what is this? We are talking about the history of understanding. One guy comes and he tells that, see, something is released by, when you look at the Parsley's experiment, okay, Joseph Priestley's experiment, he told that something is released by this plant which is filtering the air, which is not making the air poison, and he was not able to conclude what. Correct, right? And I forgot one more. And then we have Egan-House. Egan-House says that sunlight is very much essential for the plants, to undergo photosynthesis. When there was no sunlight, there was no photosynthesis. And when there was no photosynthesis, these guys died. So that's what he concluded. Then Shas, what he told? He told that plants, when they go under photosynthesis, they produce what? They produce something called as glucose, and the glucose is stored in the form of starch. Okay. Next, what happened? Eagleman comes and he says that, see, not all of the light is used by the plants. Only some part of the light is used by the plants. For that, he uses a prism experiment and he uses algae. Which one? Cladophora. Remember these guys, I'll just summarize this, but you have to make sure that it sits in your memory until your end of your exams. Okay. And he tells that out of all light, not whole light is being used up. They don't prefer whole of the light. They go for some of the of wavelengths. What wavelengths are they choosing? They choose the red wavelength and the blue wavelength of the whole sunlight. Okay. And now comes the next, breakthrough. The next breakthrough is that we understand that plants have something called as chlorophyll A and chlorophyll B. By the mid 19th century, what will happen, guys? Finally, we could understand how the photosynthesis happened through an equation. So they derived an equation, and the general equation or the empirical formula for the photosynthesis was derived by the end of 19th century. And this happens to be the empirical formula. What does it say? It says that 6 carbon atoms, 6 carbon dioxide molecules are required in the presence of 12 molecules of water, in the presence of light and chlorophyll. What will happen? You have something called as production of carbohydrate, which is a starch or glucose. And the byproducts are water as well as oxygen through this process. So that's what was understood by the end of 19th century. Now, what I want you guys to understand, hold on, I want to highlight somebody. Here comes, where is it? Yeah. See, Van Niel. Van Niel is a very important person. Please make a note of this person. What he says is, he tells something very, very important and very, very significant to understand photosynthesis. What does he suggest? He says that whatever I told in the initial time discussing the photosynthesis, he elaborated it. Let's discuss this. Okay. I hope you're understanding, right? So this is the initial thing. So we'll be rough. After that, we will get to adjust to the class. Okay. So listen, we know that carbon dioxide is required for photosynthesis. And we understood that water is also required. See, right? Water is, Van Niel is one of the guys who told that water is very much important for the process of photosynthesis. And he did not stop there. He said that, see, it's not compulsory that you always require something called as H2O. It can be H2S, or it can be HNO3, or it can be, H2SO4, sorry, H2SO4, or phosphoric acid. Basically, these are the basic things that all the organisms use for the manufacturing of energy. So what he said was, when the carbon dioxide is there and sunlight is required for breaking the bonds between the hydrogen and the corresponding molecule, when this chemical reaction is happening there, it happens because the plants require something called as electrons. So they act as electron donors. Who act as electron donors? Water, H2S, they act as electron donors. So in all case of producing energy, it's not always we get something called as O2. If the chemosynthetic bacteria, if you see, we don't get O2, we get something called as, where is it, guys? Yeah, instead of H2O, we get something, we get H2O, and what do we get here? We get sulfur. But if you look at the regular photosynthetic reaction, what do we get here? O2. You understand my point? So you remember this guy, this person, Van Niel. Van Niel, what he did? He elaborated the formula telling that hydrogen with the corresponding molecule, whether it is oxygen or sulfate, so when they split up, they produce something called as electrons. So that is why this reaction is happening. So that is a very, very, very important contribution done by, Van Niel. So now we have understood the early experiments of, how people develop over the years the understanding of how the photosynthesis works. Now let us get to the real deal. How do photosynthesis really happen, or where does the photosynthesis really take place? Now, if you look at the anatomy in flowering plants, like if you go back to the anatomy in flowering plants, we understand that leaves are one of the tremendously beautiful structure. And in leaf, apart from all the other cells that support the leaf, we have some important cells called as mesophyll cells. So mesophyll cells are a special type of parenchymatous cells. The role of mesophyll cells is to produce energy. And they do that by housing, or they are the house for a lot of chlorophyll or chloroplast. So what is a chloroplast? I will tell you an interesting story. If you look at a chloroplast and if you look at a mitochondria, what's one thing that is common, guys? What do you think about? Like you studied the cell, the structure, the unit of life, right? Cell, the unit of life. When you study this chapter, what is the speciality of chlorophyll? If you don't understand, or chloroplast, if you don't understand, or if you're not able to relate chloroplast, I will ask you something else. What about mitochondria? If you observe mitochondria, what is something very unique about it? Can anybody tell here? You can use the chat, guys. Okay, you can just communicate. Please feel free. Yeah, since we have a little bit participation in the classroom with respect to attendance, now I would want to know if you guys are really following. What is so special about mitochondria? You have so many cell organelles. For example, you have Golgi apparatus, you have ribosomes, right? And then you have endoplasmic reticulum. But something is not same when you compare mitochondria or chloroplast with other organisms, with other organelles. Anybody? Somebody? Hmm. Nobody, guys? So, cool. I will give you time. Okay. Maybe when I tell the story, you just ask me something curious. Okay. We'll discuss something curious here. See, if you look at mitochondria and if you look at chloroplast, what you can see is they are both double membrane bond organelles, correct? No, they are both double membrane bond organelles. The number of organelles do not makes something unique. What they are is they have their own genetic material in the form of DNA, correct? No, if you see in mitochondria, they have what? In mitochondria also, it has its own DNA. In chloroplast also, it has its own DNA. See, none of the cell organelles have their own DNA. DNA is like a master of the cell. It controls all the activities of the cell. When you have your own DNA, that means you can control yourself. You need not depend on the cell. So how does it even happen? So if you look back the history of how the cells were created or how the cells evolved, mitochondria and chloroplast were separate organisms itself. Okay, so they are very separate organisms itself. That's why if you look at in your cell chapter, we call these organelles as semi-autonomous. Hold on. Semi-autonomous organelles. Semi means half. Autonomous means I can govern myself. I can take care. I run my own government. I don't listen to the cell completely. I listen, but I involve in the cell functions, but I don't listen to it completely because I can do all the things that any cell can do. So these two organelles are very, very unique. So because they say that during the course of evolution, mitochondria and chloroplasts were separate organisms. So somehow they got integrated into the main cells. So this is the main cell. They told that, guys, may I come in? So they let them in. Okay, they wanted protection. They wanted other things too. So they came into this cell and they got incorporated. So if you look at your body, each and every cell is living in a symbiotic relationship with mitochondria, which is so beautiful and such a story to come across, right? We don't realize that they can be a separate organisms itself until we understand their nature. Okay, so chloroplasts are the sites for the photosynthesis, and N. Grew or Neil Grew were the first ones to study. Okay, along with Schaas, Mayer, and Dochnet. Okay, and they studied this chloroplast. So if you remember the structure of chloroplast, you know that chloroplast is a double membrane. You know, a double membrane and structure or double membrane cell organelle, I can tell. Correct? No. So if we hold on, do I have a good picture of chloroplast? No. Sure. See, look here, guys. I want you to concentrate here so that we will understand the further things. We have the outer membrane and we have the inner membrane. And between this membrane, there is a space, correct? No? This is the outer membrane of the chloroplast. This is the inner membrane of the chloroplast. Between this membrane, there is a space. And this space is called as periplastid space. Okay? So this periplastid space is very, very important to conduct so many activities of the chloroplast. Anyways, within this chloroplast envelope, what do we have is we have something called as stroma. Stroma is a mat, let's say that it's like a jelly. Okay, stroma is what? It's like a jelly. So this particular stroma is completely occupying the whole space of your whole inner space of your chloroplast. Got it? The complete inner space of your chloroplast is filled by what? Stroma. And if you observe the stroma carefully, what you can see is there are some coin-like structures piled up, and each pillar-like structure, let's say that there are like pillar-like structures, each pillar-like structures will have coin-like structures. If you place the coins one upon each other, I think you already know this, right? So these pillar-like structures, you can see this, these coin-like structures are piled up like this into small, small pillars. Let's say that each of this pillar is called as what? It's called as a grana. Okay, it's called as a what? Grana. And each coin-like structure that we have here, it is called as a thylakoid. You understand this point? So there are 200, around 200 coin-like structures are piled up with each other, and each pillar is called as a grana, and each coin-like structure is called as a thylakoid. Remember this. Now, why this thylakoid is coming here? This coin-like structure is no regular coin. I mean, this is no regular structure. It has a lot of functions to do. So if you look at each coin-like structure, that is your thylakoid, what you can see is there is a membrane. There is a membrane. So we call it as thylakoid membrane. Okay. We can call it as thylakoid membrane. So this thylakoid membrane, within this thylakoid membrane, what is there? There is a space. And what do you call this space? Lumen. We call it as lumen of thylakoid. Did you guys understand? You can see very clearly here. I hope you're following me. If you don't follow me, just please let me know in the chat box. Got it? So we have a thylakoid membrane. Within the thylakoid membrane, there is a space. We call it as a thylakoid space or a lumen of a thylakoid. There is a reason why I'm telling so detailedly about everything. Got it? So now, this is one pillar and this is another pillar, let's say. Guys, listen to me. This is one pillar and this is another pillar, let's say. Now, connecting these pillars is like a membrane-like structure. It's like a membrane just connecting like a bridge. Okay? A flat membranous structures. And this flat membranous structures are called as stroma lamella. Okay? Or it is also called as stromal thylakoids. Okay? Here it is. They're called as what? Stroma lamella, or it is also called as stromal thylakoids. Now, even stromal thylakoids, they also have a membrane. And within that membrane, what do you have? You have stroma, lumen of stroma lamellae. We can call it as lumen of stroma lamellae. Now, what do you have to remember? You have to remember something like this. We have a chloroplast. Chloroplast is a double membrane system. We know that. And within the double membrane system, we have outer membrane, we have inner membrane. Within this membrane, there is a periplastid place, cool. Now, what is within the membrane? Within membrane is a jelly-like structure occupying the whole space. And this whole thing is called as what? The stroma. Remember this. This whole thing is called as what? Stroma. If you observe what is in the stroma, in the whole room, what is present? There are coin-like structures piled up. So these whole pillar-like structures are called as what? Grana. So it's a plural. So if you observe each grana is made up of what? Piled up coin-like structures, and we call them as what? Thylakoids. Now, the important point here for us is we have to observe individual thylakoids. If you observe individual thylakoids, what do we have here? In individual thylakoids, we have a thylakoid membrane. A membrane is surrounding this whole coin-like structure. And within this membrane, what do we have? We have a lumen, a space within this. So this space is called as what? Lumen of thylakoids. Got it? And connecting these piles, coin-like piles, are called as what? Stroma lamella. Stroma lamella is different and stroma is different. That's why I want you to, have the clarity about it. Stroma is different. Stroma is the whole space. Stroma lamella is a bridge between the two grana-like structures. Is it clear, guys? Yes or no? Are you following me? Can you give a thumbs up or tell that yes, no in the chat box? It will be very encouraging, guys. Okay. Usually you communicate with me, but today is exceptionally quiet. Yeah, perfect. See, voice is not audible. Is it the same for everybody, guys? Please, somebody has a technical problem. I want others' confirmation. Hold on, somebody is texting. No, I think there is a network issue with you. Okay, please check out whoever has the voice issue. And others, did you understand? Like, are you following me? Can I move on? If you don't understand this much, it will be difficult for you guys to go ahead and understand what's the process. Clear? Chalo. I'll move on. If you have doubts, we can repeat this. So once you are aware of these major structures, remember, they usually ask what kind of genetic material is present within the chloroplast. So you should tell that it is double-stranded circular DNA. So double-stranded, you know that there are two strands involved in the DNA structure. Circular is coil, like a coil, okay, circular DNA. And along with the DNA, what do they have? They also have their own ribosomes. They also have their starch granules. They're like food granules are present, starch granules are present, okay? So this is a structure of a chloroplast. And chloroplast is a site for, the photosynthesis. What? No, they're saying that voice is clear, okay? So don't have the misunderstanding. Be polite with each other, guys. It's very, very important, okay? Chalo. So, see, this disturbs, okay? Chalo. Where were we? We understood the structure of chloroplast. Now, see, guys, if you look at the whole chloroplast, what you can see is we can see the plants with different colors. Some plants are orange, some plants are green. Most plants are green. Or some plants show the yellowish tinge, and some sea plants show a different color itself. So why the color comes is because of the pigments, like how our blood has a pigment, even they have a pigment, and we call it as chlorophyll pigments. So if we subject this whole grinding of the leaves and we put it to something called as chromatography, a technique to separate pigments. Okay, so what you can see is the what you tell that chlorophyll is one pigment that is responsible for the photosynthesis. That is not actually true. There are many other pigments that are also responsible. For example, they have chlorophyll pigments, which are chlorophyll A, chlorophyll B, carotenoid pigments, and you have xanthophyll pigments. Okay, xanthophyll pigments. So all these pigments are involving actively in the process of photosynthesis. You clear? So now, where does the photosynthesis happen, or how does the photosynthesis happen? We know that pigments are something that is very much involved in the primary function of the photosynthesis. What is their function? Let us assume that you have your pigments, and our major pigments are who? Chlorophyll. Chlorophyll is our I'm sorry if I'm yeah, chlorophyll is our major pigment. What are the types of chlorophyll we have? We have something called as chlorophyll A and chlorophyll B. You see, chlorophyll A and chlorophyll B are very major pigments. Apart from this chlorophyll A and chlorophyll B, we have accessory pigments. What are the accessory pigments? Your carotenoids, your xanthophylls, okay, are all coming under accessory pigments. What they do is, let's assume that we have chlorophyll A here. They surround this chlorophyll A, continuously. These pigments will continuously surround your chlorophyll A, and they create something called as an antenna. So they create something called as what? An antenna. The whole structure is called as a light harvesting system. So this pigment arrangement is called as what? A light harvesting system. So how are they arranged? You have a very important pigment, that is your chlorophyll A, okay? And surrounding this is your accessory pigments like chlorophyll B, carotenoids, and xanthophyll, and they surround it very, very thick, okay? So they surround it very, very thick. What is the reason? The reason is, if you look at your chlorophyll A, the chlorophyll A is a very, very, very important pigment. That's why we call it as a reaction center. So we call it as a reaction center. Sorry, we call it as a reaction center. Okay. And chlorophyll A is a reaction center. What about the rest of them? The rest of them act like something called as antenna. I told you, no, they act something like an antenna, like a dish. So together with your chlorophyll A and with your accessory pigments, they create a harvesting system. What are the harvesting? Light harvesting system they make. Now, if you look at the pigments, I told you the major reaction is happening at chlorophyll A. And the chlorophyll A, what it does is it does not absorb all the light. So it is sensitive to a particular wavelength of light. So what is a particular wavelength of light that it absorbs? It absorbs, it absorbs 680 nanometer, and 700 nanometer wavelength of lights. Sorry, yeah, 700 nanometer of wavelength of light. So whichever the chlorophyll pigments or whichever the light harvesting pigments that has the capacity to absorb 700 nanometer wavelength of light, they are called as photosystem. Sorry, here it should be. Here it should be. I'll just erase this, guys. Hold on. I will make it more clear for you. This is 700 nanometer. This is 680 nanometer. So that it will be very clear for you guys. Okay. Now, what is happening? Chlorophyll does the job, and it does the job not by absorbing all the light. There is only one particular wavelength of light that these plants use. And we call this as PAR radiation. What do we call it as? PAR radiation. What is PAR radiation? Photosynthetic active radiation. Okay. Photosynthetically active radiation is the radiation that out of all the light that is present, only a particular part of light these plants are using. Okay. So what part are they using? We can just narrow down this whole thing into two major wavelengths. What are those? One is a 680 nanometer wavelength. Other one is a 700 nanometer wavelength of light. We can divide major wavelengths. So now, whichever your light harvesting system has your chlorophyll A capable of harvesting 700 nanometer wavelength of light, they are called as photosystem 2. If they are capable of harvesting only 680 nanometer of wavelength of light, they are called as photosystem 1. Is it clear, guys? Yeah? Oh, yeah. Is it clear? So, come again. I was not able to hear you. I could not get you. Okay. So before I move on, I will just try to launch a true or false poll. If you understood, put it as true. If you did not understand, put it as false, so that I will understand what's going on, guys. Maybe you're very shy to just communicate. Just put it here. If you understood, put it as true. If you didn't understand, put it as false, so I can repeat. Yeah. Okay. Many people didn't understand here. Let's do this. Okay. Okay. Fine. That's good. That's good. So let's get back to where we started. See, our story started with photosynthesis, and where it happens, we understand that it happens in the leaves. In the leaves, where we have mesophyll cells. So, in the mesophyll cells, what do we have? We have special organelles, cell organelles called as chloroplast. And we understood the structure of a chloroplast very clearly. Now, we understand that the chloroplast has something called as pigments, correct? No? We have been studying that chloroplast contains pigments, and these pigments are called as chlorophyll pigments. But it is not completely true. If you observe what are the pigments that are present in your leaf, you come across major pigments like, I need some space. Okay. I'll write it here only, guys. I cannot find some space. Okay. I'll write it here only. Where did it go? Yeah. We have chlorophyll A, chlorophyll B, and then we have carotenoids. We have xanthophyll. Okay. Xanthophyll. This is the whole pigments that are present. Okay. Now, what happens is your chlorophyll A is there. No, this chlorophyll A is a very important pigment because it does major job. It does the major job in the process of photosynthesis. So what job does it do? This chlorophyll A, it is called as a reaction center. Reaction center. This pigment alone is called as a reaction center for a particular reason. Now imagine this reaction center, a chlorophyll A is present here. This is your chlorophyll A. And surrounding your chlorophyll A is your different pigments. What are those? Here comes your chlorophyll B, the carotenoids, the xanthophyll, they all surround it and they create an antenna-like structure. Okay. So this whole thing, you see, the surrounding pigments are called as what? Antenna. Okay. The inner thing, the core is what? It is chlorophyll A. What is it called as? It is called as reaction center. Combining both, what do we call them? We call them as light harvesting systems. So if you observe your chloroplast, okay, you come across two important light harvesting systems. What are those? One is the photosystem 1, other one is called as a photosystem 2. So what is photosystem 1 and what is photosystem 2 and what are the differences between them? If you see the whole light, see this is your whole light, correct? No? If you split the light, you come across this much like this. You can get a whip jar. And here, the light can be coming from 400 nanometer to 750 nanometers. That is the wavelength of your light, right? Different wavelengths have what? Different rays. For example, 400 nanometer have gamma rays. Similarly, you have radio waves and infrared rays. If you look at plants, plants utilize a certain wavelength of light for manufacturing the solar energy. And this we call it as PAR radiation. Okay, we call it as PAR radiation. And this PAR radiation, what is this PAR radiation and what wavelength are they manufacturing food? Now let's come back to this harvesting system. We will understand better. So if you see that chlorophyll A has the capacity to harvest two different types of wavelengths, major wavelengths. What are those? One is 700 nanometer wavelength light. Other one is called as 680 nanometer wavelength light. Got it? So some chlorophyll A is dedicated to harvest 700 nanometer wavelength of light, and some chlorophyll A are dedicated to harvest 680 nanometer wavelength of light. Got it? If you have a light harvesting system with your chlorophyll A, okay, with your chlorophyll A, okay, here also there is chlorophyll A, okay, here also there is chlorophyll A. Imagine that one chlorophyll A has a light harvesting system with 700 nanometer. So this chlorophyll A has a capacity to harvest how much? 700 nanometer wavelength of light. And another chlorophyll A in another light harvesting system harvest only 680 nanometer wavelength of light. So if you have any light harvesting system with chlorophyll A with 700 nanometer wavelength absorption capacity, we call them as photosystems 2. We call it as photosystem 2. And then we have photosystem 1. So any light harvesting system with your chlorophyll A having the capacity to harvest only 680 nanometer wavelength of light, it is called as photosystem 1. So when you come across photosystem 2, remember that it is 700 nanometer. And when it comes to photosystem 1, you remember that it is 680 nanometer wavelength of light. Clear? So this is the job of only chlorophyll A. I told you that light harvesting system has one chlorophyll A and rest of it is accessory pigments. What are these accessory pigments are doing? The accessory pigments, they harvest different frequencies or different wavelengths of light. One thing is they go and explore different wavelengths. For example, they explore 500, 550 nanometers, 450 nanometers, all these different nanometers of lights which comes incident to the plant, they absorb. Okay, they will try to do their job, give their contribution. So that is one thing. They absorb other frequencies except 680 and 700 nanometers. Another job is they protect your chlorophyll A. How do they protect this chlorophyll A? By a process something called as photooxidation. I'll explain you what is photooxidation as we move further. Clear, no? You understood what is, see, what did you understand? You understood what are pigments, what are the different types of pigments, how are the pigments arranged, what is a major pigment, chlorophyll A, what is it called as reaction center, and accessory pigments are surrounding, what are they called as antenna. So your reaction center, that is your chlorophyll A, plus all your accessory pigments together, they are called as what? Light harvesting systems. So in that light harvesting system, we can come across two types of light harvesting systems. What are those? One is photosystem 2 and photosystem 1, or photosystem 1 or 2. How do we differentiate? If you have a chlorophyll A with having the capacity of 700 nanometer light harvesting, we call it as photosystem 2. If it is photosystem 1, they have the chlorophyll A that is harvesting 680 nanometer wavelength of light. Now, I will go on to add a little more very important information. Where are these pigments present? These pigments are present in your thylakoids, major, right now. So, your thylakoids, especially in the membranes. Thylakoid membranes are there, no? I told no. These coin-like structures, they have a membrane. In this membrane, you have your pigments, correct? Now, if you look at the membranes of the thylakoids, what you can see is you can come across both the photosystems. They have both photosystem 2 as well as photosystem 1. Thylakoids have both light harvesting systems. Whereas, if you look at another structure, what is another structure? Stroma lamella. Okay, your stroma lamella, this bridge-like structures, I told you, these bridge-like structures that are present between two granas. So these bridge-like structures, which are called as stroma lamella, they have only one type of photosystems. Very, very important information. What type of photosystem only present in stroma lamella? It is photosystem 1. Okay, it is called as photosystem 1. Got it? No? This particular photosystem 1 is present both in thylakoids as well as stroma lamella. But if you look at photosystem 2, they are not present in your stroma lamella. They're only present in the membranes as well as in the thylakoids. Okay. Now, did you understand this part? Guys, give me an answer now. Should I launch a poll again? True, false? So it will be easy for you. Come on. The basics are very important, guys. Rest of the mechanical process, it's very, very simple to understand. If you don't understand where it's happening, so it would be really, really hard to go ahead. Okay. So the two people who are telling that you don't understand, like somewhere you have missed out, please put your doubt. Okay. So that I can be specific, because I repeated very slowly, I feel, and I feel I did a little bit of justice for the, for what you have asked. Now I'm not sure where you have missed. Instead of repeating the whole thing, please point out this is where I have missed. Got it now, guys? Yeah. So now what happens is you have your whole system. You see that this is the organelle that is responsible for this particular process. We understood that the pigments are responsible. We understood how the pigments are organized, and we also saw what do these pigments do? They harvest light. Is it all of light? No, they specifically harvest a particular frequency of light. Now, what is the role of these pigments? What are they involved in? They are involved in something called as, yeah, they are involved in completely light based reactions, correct? No, they are involved in light dependent reactions. So when they're involved in light dependent reactions, what do is they use the light, who these pigments use the light with other compounds like carbon dioxide, water. So finally what they do is when they use this light, so these pigments, the reaction centers in these pigments, their role is to make sure by the end of the process, they produce ATP, which is a source of energy, and another very energy rich, very high energy rich molecule called as NADPH, which are called as light reactions. Light reactions are the reactions which are dependent on light, and these pigments and these organelles are very much involved in it. Is it clear? Okay. So what is the end? What do they do? What is their job? Their job is simple. They take the light, they split the water, whatever it is present there. They use all the raw materials. By the end of their role is to produce two important components. What are those? One is ATP, other one is called as NADPH. As simple as that. Okay. Chalo. Now you can see these are all the pigments, guys. I should have discussed this very detailedly, but it's all right. You can see, I will just go through this. You can see the chlorophyll A, which is a very, very important pigment. Okay. So now these particular chlorophyll A, chlorophyll B, you have chlorophyll C, D, E, and you have bacteriochlorophyll in some of the purple sulfur, very, very important. We call it as purple sulfur bacteria, which is your, what do you tell, archibacteria, right? So they also called as bacteriorhodopsin. So they do not convert to any other byproducts. Like, for example, if you see the whole autotrophic reactions, you can tell that plants produce something called as glucose. Correct? No, by the end of photosynthesis, you can see that they produce glucose. And they break down this glucose to produce what, guys? ATP. Correct? No, there is a whole chain of process to get energy. But bacteria having bacteriorhodopsin, they don't do this. What they directly break down these chemicals and convert this into direct energy. They only get ATP. There is nothing called as formation of glucose. So these are the unique things. Then you have other pigments called as carotenoids, and then you have carotenes and xanthophylls. So these are the pigments which are accessory pigments. And what is our major pigment? Chlorophyll A and chlorophyll B. Major, very, very major pigment, chlorophyll A. Clear? So this is done. Now, immersion effect, we will talk about it, okay? And we'll see how this immersion effect will give us a proof that a particular frequency of light is very, very important for increasing the rate of photosynthesis, okay? So that is secondary. Now, the point is we are almost out of time. So I have to push forward and move faster. But anyways, today things got a little late. I took it very, very slow as well. All right, guys. Thank you very much for coming. And 5 minutes is there, and I'm ending it for a reason, because I have to start with light reactions. That is why if you have any doubts, please let me know. Or if you have doubts further to the chapter, you can ask me 5 minutes. Okay? I can't hear you guys. I can't hear maybe something they're talking. All right. So here is a poll. Please give a class feedback poll because I told you already, guys. Hold on. Guys, can you please mute? Excuse me. Okay, hold on. I will mute you guys. Just be in the class, guys. Just 1 second. They're talking in my mother tongue. All right. I've muted all. So I want your poll, guys. Please give me that. We have started a completely new topic, no? So some of you might have not caught, and there is no time for catching up. Okay, that's cool. Give you a honest review. We can just correct this. Okay. And there is no only yes, no, guys. What's happening here? Yeah, again, no. Now I should ask what? No. Now I should ask you guys, right? What is the meaning of no? Chalo. So if you have any doubts, please ask because these chapters, it might look very lengthy, very dry and hard to remember, but trust me, easy scoring. Botany people are kind enough. Okay. They give very direct questions. All right. So please ask your doubts. I'm here and don't hesitate. And meanwhile, I will end my stream. And please come on time. No, we already have less classes. Uh-oh, I think this is the most unusual poll I ever got. Cool. I respect that. Thank you for the honest review, guys. Some people think that it was not easy to understand the concept and moderate. Some people tell that, guys, you didn't ask me the question. Okay. You should ask the question in the chat. No, then I will be able to answer. Next one is how confident are you? Okay. That is left to you guys. So many people felt the pace is normal. Overall session, you are rating on an average of 3. I don't blame you guys, but come tomorrow. We will go in detail and very little bit faster. Hold on. Let me check your questions also. If you have some. Okay. ಊಟ ಇಲ್ಲ. Okay. All right, guys. You are up for the next session. You can go and attend, right? And you can leave the class, okay? Because it will take a little bit time for me to end the session. ಏನ್ರೋ ನಿಮ್ದು ಇಲ್ಲಿ ಬ್ರೋಸ್ ಚಾಟ್ ಮಾಡ್ತಿರೋದಾ ನೀವು? ಜಾಸ್ತಿ ಜನ ಇರ್ತಾರೆ ಕ್ಲಾಸ್ ಅಲ್ಲಿ, ಸೊ ಯುಟಿಲೈಸ್ ಪ್ರಾಪರ್ಲಿ ಗೈಸ್. How you behave will affect the future classes also, right? So at least one student is definitely getting benefited out of it. Respect their time and energy. ನೀವು ಮಾಡೋ ತರಲೇ ಎಲ್ಲ ನಾನು ಬೇಡ ಅನ್ನಲ್ಲ, ಬಟ್ there's a place, no? So it will get reflected. Everything is recorded. So it brings a bad name for the JNV as well as for your particular JNV. And it also shows the seriousness of the class. It's like directly or indirectly, you are not humiliating, right? So it's not okay, guys. Otherwise, I'll give, right? I'll give the leverage for you guys to express, but it's not the place. Please understand. Okay. Chalo. Bye, guys. Have a nice day and take care. I'll see you in the next class. So it just reflects what you are, man. Totally. The others you can leave. And maybe this guy's having fun. Let me see about how far it goes, ಉತ್ತರ ಕನ್ನಡ ಐ ಥಿಂಕ್ ನೀವೆಲ್ಲ ಇದೆ ಅನ್ಸುತ್ತೆ ಕನ್ನಡದವ್ರೆ ನೋಡಿ ಪರವಾಗಿಲ್ಲ.